Okay, let's unpack this. Have you ever found yourself needing to quickly get up to speed on some complex biological information, maybe for a medical meeting, an exam, or just because your curiosity about the human body at a microscopic level is absolutely insatiable?
Yeah, that feeling of needing the core info, like now.
Exactly. Well, you were definitely in the right place. Welcome to the deep dive. Today, our mission is a fascinating plunge into the foundational world of epithelial tissue.
And we'll be drawing our insights from the really comprehensive Histology, a text and atlas, 8th edition. Our goal is to pull out the most important nuggets of knowledge from this text.
Right. Focusing on things like the microscopic anatomy, the cells themselves, different tissue types.
And those crucial clinical correlations, basically to give you a real shortcut to being, properly well informed on this.
When we talk about how our bodies are built, we often think about the big stuff, the organs, right? But it all starts with these incredible tiny building blocks, tissues. And today, we're zooming right in on epithelial tissue, which is, well, it's pretty much everywhere. Think of it as the body's natural lining protecting you, letting things be absorbed, even secreting essential stuff.
And what's really fascinating, I think, is how these layers, which seem quite simple on the surface, perform such diverse and absolutely critical functions. We'll try to connect these tiny microscopic details to their real-world impact on human health. It highlights some really elegant design principles in the body.
Okay, so let's start right at the beginning. What exactly is epithelial tissue? Lay it out for us.
Great question. So, at its core, epithelial tissue is this specialized tissue that covers all our body surfaces, and it lines various internal cavities, too. It's unique because it's avascular.
Avascular, meaning no blood vessels.
Exactly, no blood vessels of its own. It gets its nutrients by diffusion, basically soaking them up from the connective tissue underneath.
Right.
And this tissue forms a selective barrier. It controls what gets to pass between the environment, could be internal, could be external, and the rest of the body. And some specialized epithelial cells even act as receptors for our senses, like, taste or smell.
Wow, that's a huge set of jobs for what sounds like a pretty thin layer. So how do these cells actually manage it? How do they create such a powerful barrier and do all these different things?
Well, it really comes down to their fundamental characteristics. First off, epithelial cells are incredibly tightly packed. They stick to one another through specialized connections called cell junctions.
Like a really tightly woven fabric.
That's a great analogy. Almost no gaps. This close packing is just essential for their barrier function, and critically, they have polarity.
Polarity. What does that mean for a cell? Sounds kind of directional.
It is. It means different sides of the cell have distinct structures and therefore different jobs. You've got the apical domain, that's the free surface, facing the outside world or maybe a body cavity like the inside of your stomach.
Okay, the top surface.
Right. Then there's the lateral domain, which is, on the sides, communicating with the cells next to it. And finally, the basal domain, the bottom, which attaches to the underlying support structure. That's called the basement membrane.
Ah, the basement membrane.
And the specific proteins and lipids, the molecules on each of these surfaces, give them their unique properties and functions.
So this basement membrane, is it actually part of the cell itself or something else?
Good question. No, not exactly part of the cell. It's a non-cellular layer, rich in proteins and polysaccharides, that the basal surface of the epithelial cells sticks to really securely.
Like a foundation.
Exactly. Think of it as the foundation or the glue holding the epithelial cells in place. And you can actually see it under a microscope if you use special stains like PAS stain, which makes it show up magenta.
Fascinating. So we've got these tightly packed polarized cells stuck onto a basement membrane. How on earth do we start classifying them? There must be loads of different types.
There are, but our source, the textbook, gives us a pretty straightforward way. It's based on two main things: the number of cell layers and the shape of the cells on the surface layer.
Okay, number of layers first.
Right. Simple epithelium is just one cell layer thick. That's perfect for processes like absorption or filtration, where things need to pass through easily.
Makes sense. Thin is good for transport.
Exactly. Then you have stratified epithelium, which has 2 or more layers. This makes it much tougher, ideal for protection in areas that get a lot of wear and tear, like your skin.
Okay, simple and stratified. What about the shapes?
For shape, we look at the surface cells, especially in stratified types. You've got squamous cells. They're flat, wider than they are tall, almost like scales or paving stones.
Allotments, got it.
Then cuboidal cells, which are pretty much cube-shaped. Their width, depth, and height are roughly the same.
Like little boxes.
Yep. And finally, columnar cells. These are taller than they are wide, like columns.
Okay, squamous, cuboidal, columnar.
And then you just combine these. So you get simple squamous, really thin, found-lining blood vessels, where we actually give it a special name, endothelium.
Ah, endothelium. I've heard of that.
Or you have stratified squamous, which is your skin's outer layer, the epidermis, very protective. Then simple cuboidal in kidney tubules, simple columnar lining your stomach. You get the idea.
Yeah, mixing and matching layers and shapes.
We also have a couple of special cases. Pseudostratified epithelium looks stratified, kind of messy under the microscope.
Pseudo meaning false, right? So it looks layered but isn't.
Exactly. All the cells actually touch the basement membrane, even if they look stacked. Your trachea, your windpipe is a classic example. It often has cilia on top, too.
Okay, that's tricky.
It can be. Identifying it often relies on knowing its typical location. And then there's a really cool one, transitional epithelium, also called urothelium.
Urothelium. Sounds like urinary system.
Precisely. It lines your lower urinary tract bladder, ureters. What's amazing is its ability to stretch. When your bladder is empty, the surface cells look large and kind of dome-shaped, puffy almost.
Right.
But as the bladder fills up with urine, the tissue stretches, and those surface cells flatten out, becoming more squamous-like to accommodate the volume.
That's incredible. Talk about structure-fitting function.
Absolutely. It's an elegant example. And think about that simple squamous endothelium lining your blood vessels. It's incredibly thin, but so specialized. It regulates blood flow, immune responses, all sorts.
Yeah.
And the mesothelium lining your closed body cavities, like your abdominal cavity, does similar protective and lubricating jobs.
So if epithelial tissue is basically the body's skin at every level, internal and external, how do different sides of the same cell manage to do different jobs? You mentioned polarity.
Right, that polarity is key. It means the cell has distinct domains or surfaces, and each has specialized machinery. Let's start with the apical domain, the top or free surface.
Okay, the side facing the outside or the lumen. What's special there?
Well, it's often equipped with some fascinating structures, modifications that help it do its job. The first common ones are microvilli.
Ah, the microvilli. I remember these. They're like tiny finger-like projections, right? Increases surface area.
Exactly. They dramatically increase the cell's surface area. Think of them like thousands of microscopic bristles on a brush border. This is absolutely crucial for cells that are heavily involved in absorption.
Like in the gut.
Perfect example. The cells lining your small intestine need massive surface area to absorb nutrients. Same for cells in your kidney tubules reabsorbing useful molecules. These microvilli have an internal framework of actin filaments, strong proteins anchored into the cell.
Okay, so microvilli boost absorption. What else is up there?
Then you have something called stereocilia. Now, despite the name cilia, they're actually unusually long, immotile microvilli. They don't move.
Wait, stereocilia aren't actually cilia? That's confusing.
It is a bit confusing. Think of them as super long microvilli. You find them in the male reproductive system, in the epididymis, helping with absorption. But their really remarkable role is as mechanoreceptors in the sensory hair cells of your inner ear.
For hearing.
For hearing and balance, yes. They are exquisitely sensitive to vibration and movement, and they have this amazing internal rebuilding process, constantly adding and removing protein subunits to maintain their precise length over your lifetime. Super important for function.
Wow. So immodal stereocilia. Are there modal ones too? Actual cilia.
Yes. Then we get to true cilia. These are longer, hair-like extensions that can move. They have a complex internal structure based on microtubules, kind of like internal scaffolding.
Okay, and these are the ones that sweep things along?
Most of them, yes. The modal cilia typically have a distinctive 9+2 arrangement of microtubules inside. That's 9 pairs arranged in a circle around 2 central ones. Critically, they contain a motor protein called dynein.
Dynein.
Yep. Dynein uses energy, ATP, to make the cilium bend and create a coordinated sweeping motion. You find hundreds of these on cells lining your trachea and bronchi.
Clearing out mucus and dust.
Exactly. They rhythmically sweep mucus and trapped particles up and out of your airways. You also find them in the female reproductive tract, in the oviducts, helping transport the egg.
Okay, this makes me think, if these are so important for clearing airways, what happens if they don't work properly?
That brings us to a really powerful clinical connection. Primary ciliary dyskinesia, or PCD, sometimes called immotile cilia syndrome. It's a genetic disorder often involving a defect in those dynein motor proteins.
So the cilia can't move or can't move effectively.
Right. And the consequences are significant. Chronic respiratory problems, recurring infections, bronchitis because mucus just sits there. Also, often male sterility because the flagella on sperm, which are basically modified cilia, can't propel the sperm.
Oh, wow. That's serious.
It is. And there's another fascinating link. About half of people with PCD also have a condition called situs inversus.
Situs inversus? What's that?
It's where the internal organs are arranged in a mirror image of the normal pattern. Heart on the right, liver on the left, and so on. Or sometimes randomly positioned.
Whoa. How is that connected to cilia?
It connects to another type of cilia, but first let's mention primary cilia. These are different. They are usually solitary, just one per cell, and they are immotile. They're found on almost every single cell type in your body.
One immotile cilium on all and every cell. What do they do if they don't move stuff?
They act as crucial sensors, like tiny antennae. They sense chemical signals, osmotic pressure, even physical forces like fluid flow. They play roles in mediating your perception of light, odor, and sound, and they are absolutely vital for normal tissue development.
Okay, sensors. Give me an example.
The key one is in your kidney tubules. The primary cilia there act as mechanoreceptors, sensing the flow of fluid passing through the tubule. This sensing triggers responses inside the cell.
And if that goes wrong, another clinical connection.
Absolutely. Here's a huge aha moment. Polycystic kidney disease, or PKD. It's a relatively common genetic disorder and a major cause of kidney failure.
PKD. I've heard of that. It involves cysts in the kidneys, right?
Exactly. And many forms of PKD are linked to mutations in genes that affect proteins found in these primary cilia in the kidney tubules. If the cilia can't properly sense the fluid flow, it disrupts normal cell function, leading to the formation of numerous fluid-filled cysts that eventually destroy the kidney tissue.
Incredible. Just from a tiny sensor not working right on the cell surface.
It really shows how critical these microscopic structures are. And it can affect other organs too, cysts in the liver or pancreas, sometimes vision or hearing problems.
Okay, so back to that situs inversus thing. How do cilia control left and right in the body?
That involves a special type called nodal cilia. These are found very early in embryonic development near a structure called the primitive node. Like primary cilia, they have that 9+0 microtubule arrangement, lacking the central pair.
So normally immotile, like primary cilia.
Here's the twist. They are motile, but they move differently, with a unique rotational sort of conical movement. And this movement generates a tiny directed fluid flow across the embryonic disc, a leftward nodal flow.
A tiny current that tells the body which way is left.
Essentially, yes. This flow is thought to trigger signaling pathways that establish the correct left-right asymmetry of your internal organs during development. If these nodal cilia are absent or immotile, like sometimes happens in PCD, this flow doesn't happen correctly and organ placement becomes random, leading to situs inversus.
Mind blown.
Yeah.
Tiny rotating hairs setting up our entire body plan. Okay. So we've got amazing stuff on the apical surface, but the cells need to stick together tightly to form that barrier. Let's talk about the sides, the lateral domain and those cell junctions.
Right. The lateral domain is where cells connect and communicate with their neighbors. It's packed with specialized junctional complexes that act like molecular glue, rivets, and even communication channels.
Okay, break them down for us. What's the first type?
The most apical ones, right near the top surface, are the tight junctions, also called occluding junctions or zonula occludens. They form a continuous belt-like seal completely encircling the cell.
A seal. So their main job is stopping leaks.
Precisely. They are the primary paracellular diffusion barrier. Think of them as the zipper on a Ziploc bag. They tightly regulate what can pass between the cells, stopping unwanted stuff like bacteria or toxins from just slipping through the gaps.
Makes sense. What else?
They also act as a crucial fence within the cell membrane itself. They prevent membrane proteins and lipids that belong on the apical surface from drifting down into the lateral domain, and vice versa. This is essential for maintaining that cell polarity we talked about.
Keeping the top stuff on top and the side stuff on the sides.
Exactly. Key proteins forming these junctions are called claudins. They form the backbone, and different claudins can actually create tiny selective channels allowing specific ions, like magnesium or calcium, to pass between cells under controlled conditions. Another key protein is occludin.
Claudins and occludin.
Got it. And you mentioned clinical connections here, too, things attacking these junctions.
Oh, absolutely. This is a major battleground. For instance, the bacterium Clostridium perfringens, a common cause of food poisoning, produces a toxin that specifically binds to claudins.
What does that do?
It essentially breaks the seal, disrupts the tight junction barrier in your gut. This leads to massive leakage of fluid into the intestine, hence severe diarrhea.
Nasty.
Very. And Helicobacter pylori, the bacterium linked to stomach ulcers, also targets tight junction proteins, weakening the protective lining of the stomach. Even common allergens, like enzymes from house dust mites, can break down tight junction proteins in your respiratory epithelium, increasing permeability and contributing to asthma symptoms.
Wow. So maintaining these junctions is critical for health. What comes below the tight junctions?
Next, we have the anchoring junctions. Their main role is mechanical stability, holding the cells together strongly and linking their internal cytoskeletons, giving the whole tissue strength and resilience against physical stress. Okay, structural support. What types are there?
There are two main types that link cells to each other. First is the zonula adherens. This is also a continuous belt, usually found just below the tight junction.
Another belt.
Yes, but its job is different. It provides strong lateral adhesion, really anchoring the cells together side by side. It connects to the cell's internal actin filament network via proteins like cadherins and catenins. Think of it reinforcing the tissue against stretching or pulling forces.
Okay, the adherens junction belt for strength. What's the other anchoring type?
The other type is the macula adherens, much better known as the desmosome. These aren't continuous belts. They're more like localized spot welds or rivets scattered across the lateral surfaces.
Spot welds. I like that image. So, super strong points of attachment.
Incredibly strong. Desmosomes provide very powerful cell-to-cell adhesion. Instead of linking to actin, they anchor to the cell's intermediate filaments, things like keratin filaments and skin cells. This creates a continuous stress-bearing network throughout the entire epithelial sheet.
Connecting the internal structural cables between cells.
Exactly. They have specialized cadherin proteins, desmogleins and desmocollins, that reach across the gap between cells and bind tightly, almost like molecular Velcro. They're super abundant in tissues that endure a lot of mechanical stress, like your skin epidermis or the muscle tissue in your heart.
Makes sense. Skin needs to be tough. Okay, so tight junctions for sealing, anchoring junctions for strength.
Yeah.
What about communication?
That's the job of the third main type, communicating junctions, usually called gap junctions or nexuses. These are fundamentally different.
How so?
They form direct regulated channels or pipelines right through the membranes of adjacent cells. They allow small molecules, things like ions, amino acids, signaling molecules like cyclic AMP, to pass directly from the cytoplasm of one cell into the cytoplasm of its neighbor.
Wow. Direct cell-to-cell pipelines. What's the point of that?
It allows for very rapid communication and coordination of activity between cells. Think about your heart muscle. All those cells need to contract in near-perfect synchrony. Gap junctions allow the electrical signal, carried by ions, to spread almost instantaneously between cells.
Coordinating the heartbeat.
Exactly. They're also crucial in other tissues for coordinating transport functions, glandular secretion, and developmental processes. Each channel is formed by two halves called connexins, meeting in the middle. And each connexin is made of 6 protein subunits called connexins.
Connexins. And I bet these have clinical links too.
You bet. Mutations in different connexin genes are responsible for a surprising number of human diseases. For example, mutations in the gene for connexin 26 are a common cause of congenital deafness.
Deafness. How does the gap junction protein cause deafness?
It disrupts the crucial recycling of potassium ions within the cochlea of the inner ear, which is essential for the function of the sensory hair cells that detect sound. Without proper connexin 26 function, the system breaks down.
Incredible. Any others?
Yes, mutations in other connexins, like C-Guys46 and C-by50, are linked to certain types of inherited cataracts. The lens of your eye has no blood vessels, so it relies heavily on gap junctions to transport nutrients and metabolites between its cells. If those junctions are faulty, the lens becomes cloudy.
Wow. It really drives home how absolutely vital these microscopic connections are for everything from hearing and sight to just holding our tissues together.
It really does, and it raises that important question. How does the body manage such intricate control over these barriers and communication lines, and what happens when control fails? We see these direct links, a specific molecule like a claudin or a connexin failing, and boom, you get a specific disease. It underscores how fundamental this stuff is.
Okay, so cells stick tightly to each other on their sides. But how does the whole sheet stick to the underlying tissue? Let's move to the bottom, the basal domain.
Right. The basal domain is all about anchoring the epithelial cell to the extracellular matrix below it, that complex meshwork of proteins and polysaccharides that provides structural support in tissues. And the key player here, as we mentioned, is the basement membrane.
Which we said isn't part of the cell, but is the foundation it sits on. How does it look structurally?
Well, under a standard light microscope, especially with that PAS stain, it looks like a distinct, sometimes quite thick, amorphous layer. But with the power of an electron microscope, we see more detail. There's a very specific electron-dense layer right next to the cell membrane called the basal lamina.
Basal lamina. Is that the same as basement membrane? The terms get confusing.
Good point. The terminology can be tricky. Basal lamina specifically refers to the layer made by the epithelial cells and some other cell types like muscle cells, where it's called an external lamina. The term basement membrane, as seen in light microscopy, often includes the basal lamina plus an underlying layer of connective tissue fibers called the reticular lamina. So basal lamina is a more precise ultrastructural term.
Okay, basal lamina made by the cells, reticular lamina from connective tissue below, together forming the basement membrane we see in LM. Got it. What's the basal lamina actually made of?
It's a complex mix, but the main components are specialized proteins. About 50% is type 5e collagen, which doesn't form thick fibers like other collagens, but instead creates a flexible, sheet-like meshwork or scaffold.
The main framework.
Right. Then you have laminins. These are large, cross-shaped glycoproteins that are crucial for organizing the whole structure. They bind to receptors on the cell surface and also to the type 5e collagen network, essentially initiating the assembly of the basal lamina. There are various types of laminin adding specificity.
Okay, collagen scaffold, laminin organizers. What else?
There are also things like nitogen or intactin, which helps link laminin and type 5e collagen, and various proteoglycans like perlecan. These proteoglycans are heavily negatively charged and hold a lot of water, helping to regulate what filters through the basal lamina, especially based on charge.
And underneath that, in the connective tissue, is the reticular lamina you mentioned.
Often, yes. This layer contains reticular fibers, which are actually made of type 3 collagen. And this reticular lamina is attached to the overlying basal lamina by specialized structures.
Are they attached? More molecular glue.
Pretty much. There are anchoring fibrils, which are made of type 7 collagen. These loop out from the basal lamina and entrap the type 3 collagen fibers of the reticular lamina, securing the connection.
Type 7 collagen. Does that ring a clinical bell?
It absolutely should. Mutations in the gene for type 7 collagen cause a severe blistering skin disease called dystrophic epidermolysis bullosa. The anchoring fibrils don't form properly, so the epidermis, the outer skin layer, literally just lifts away from the underlying dermis with the slightest friction because it's not anchored down correctly.
Horrific, just from faulty anchoring fibrils.
It highlights how critical that connection is. There are also fibrillin microfibrils involved, linking the basal lamina to elastic fibers deeper in the connective tissue. Mutations in fibrillin cause Marfan syndrome.
So this basement membrane, or basal lamina complex, is way more than just glue then. What are its main functions? Why should we care?
Oh, it's multifunctional and vital. First, yes, structural attachment anchoring the epithelium. Second, compartmentalization. It creates a boundary, separating the epithelium from the underlying connective tissue. Things generally have to pass through it, not just around it.
A controlled border.
Exactly. Third, filtration. Especially in the kidney glomerulus, the specialized basement membrane there acts as a crucial sieve and charge-selective filter for blood plasma. Fourth, it provides a tissue scaffold. If epithelium is damaged, the intact basement membrane can guide the regenerating cells, helping restore the normal structure. It also acts as a physical barrier that can slow down the invasion of cancer cells.
A barrier to tumors. That's huge.
It is. And finally, it's involved in regulation and signaling. Molecules within the basement membrane can bind to receptors on the cell surface and influence cell behavior, things like proliferation, differentiation, migration, even gene expression. It's an active participant, not just passive support.
Okay, so the cells make this complex layer below them. How do the cells actually attach to it? What are the specific junctions?
There are two main types of junctions connecting the cell's basal domain to the basal lamina. The first are focal adhesions.
Focal adhesions, what do they do?
These are generally more dynamic connections. They link the cell's internal actin cytoskeleton, those stress fibers inside the cell, to proteins in the extracellular matrix, like fibronectin and laminin, via transmembrane proteins called integrins.
Integrins. They sound important.
They are. Integrins act as the bridge. Focal adhesions are really important for processes like cell migration, for example, during wound healing, where cells need to crawl over the matrix. They're like temporary, movable, grappling hooks. They also act as mechanosensors, detecting physical forces.
Okay, dynamic links for movement. What's the other type?
The other type provides much more stable, strong anchoring. These are the hemidesmosomes.
Hemidesmosomes, like half a desmosome.
Exactly. They look a bit like half of one of those spot weld desmosomes we talked about on the lateral surface, but their structure and function are different. They anchor the cell's intermediate filaments, like keratin and skin, to the underlying basal lamina.
So strong anchoring to the base using intermediate filaments.
Right. They are crucial in epithelia that are subjected to significant abrasion or shearing forces. Think cornea, skin, lining of the mouth, esophagus. They provide really robust adhesion. Key proteins here include a specific type of integrin, IST4 integrin, and also type XF7 collagen, which is actually a transmembrane protein in this context.
And I sense more clinical links coming.
Definitely. Remember those blistering diseases. Bullous pemphigoid is an autoimmune disease where the body makes antibodies against components of the hemidesmosome, specifically proteins called BP230 and that type X7 collagen. The attack disrupts the adhesion, causing the epidermis to detach from the dermis, forming large blisters.
Autoimmune attack on the anchors.
Exactly. And another genetic blistering disease, junctional epidermolysis bullosa, is often caused by mutations in genes for laminin 332, a specific type of laminin that's essential for hemidesmosome formation and stability. Again, faulty anchors lead to tissue separation.
It's striking how many diseases trace back to failures in these specific molecular connections.
It really is. It highlights the importance of understanding this level of detail. One last feature of the basal domain. Some cells have basal enfoldings.
Folds in the bottom membrane. Why?
It dramatically increases the surface area of the basal membrane. This is really prominent in cells that are actively transporting ions or water across the epithelium, like in kidney tubules or certain salivary gland ducts. More surface area means more space for transport proteins and ion pumps. Often, mitochondria are packed into these folds to supply the energy needed, giving the base of the cell a striated or striped appearance under the microscope.
More surface area for transport makes sense.
Yeah.
Okay, so we've covered the surfaces, the junctions, the base. What about glands? You mentioned epithelial cells form glands.
Yes, gland formation is a major function, a specialization of epithelial tissue. Glands are essentially organized collections of epithelial cells, specialized for secretion making and releasing specific products.
How do we classify them?
A major way is based on how and where they release their products. You have exocrine glands. These secrete their products onto a surface, either directly onto the body surface, like sweat glands, or into a cavity or lumen via ducts, like salivary glands or the pancreas secreting digestive enzymes into the gut.
Exocrine means secreting outwards through ducts.
Right. Then you have endocrine glands. These glands have lost their connection to the surface during development. They don't have ducts. They secrete their products, which are hormones, directly into the surrounding connective tissue, where they then enter the bloodstream and travel to target cells elsewhere in the body. Think thyroid gland, adrenal gland, endocrine parts of the pancreas releasing insulin.
Endocrine means secreting inwards into the blood. Hormones.
Exactly. And there's also local signaling. Paracrine signaling is when a cell releases a substance that acts only on nearby, neighboring cells. Autocrine signaling is when a cell releases a substance that acts on itself.
Okay, exocrine, endocrine, paracrine, autocrine. Different destinations. What about how exocrine glands release their stuff? Are there different mechanisms?
Yes, there are three main mechanisms of secretion for exocrine glands. The most common is merocrine secretion. Here, the secretory product is packaged into membrane-bound vesicles inside the cell. These vesicles then move to the apical surface, fuse with the cell membrane, and release their contents outside the cell by exocytosis. No loss of cytoplasm. Pancreatic acinar cells releasing enzymes work this way.
Merocrine is just releasing the product via vesicles.
Right. Then there's apocrine secretion. In this case, the secretory product accumulates at the apical tip of the cell, and then that whole portion of the apex, surrounded by a bit of cytoplasm and membrane, pinches off and is released. Some lipid secretion in the lactating mammary gland is a classic example.
Apocrine sheds the apex of the cell.
Correct. And finally, holocrine secretion. This is the most dramatic. The entire secretory cell fills up with the product, then undergoes programmed cell death apoptosis, and the whole disintegrating cell, along with its contents, becomes the secretion. Sebaceous glands in your skin, releasing oily sebum, are the prime example. The cells literally self-destruct to release the product.
Holocrin, the whole cell goes, wow, okay, merocrine, apocrine, holocrin. And glands can be simple cells or complex structures, right?
Yes. The simplest are unicellular glands, single secretory cells scattered among other non-secretory epithelial cells. The classic example is the goblet cell found in the lining of your intestines and respiratory tract, which secretes mucus.
Goblet cells making mucus.
Then you have multicellular glands, which range from simple tubes or sacs to highly complex branched structures with elaborate duct systems. We classify them based on whether their duct is branched, compound, or unbranched, simple, and the shape of their secretory portions, tubular, acinar, like a berry or flask, or tubuloacinar.
Lots of structural variety there. What about the type of stuff they secrete?
Broadly, we often distinguish between mucus secretions and serous secretions. Mucus secretions are viscous and slimy, rich in heavily glycosylated proteins called mucins. Goblet cells are mucus, as are some salivary glands. The mucus protects and lubricates surfaces. Under the microscope, mucus cells often look pale or empty because the mucus doesn't stain well with standard H&E, and their nucleus is typically flattened at the base.
Mucus is thick and slimy. What about serous?
Serous secretions are typically more watery and rich in proteins, often enzymes. Think of the parotid salivary gland or the exocrine pancreas. Serous cells usually have a round nucleus, and their apical cytoplasm might stain pinkish if secretory granules are preserved, while the base of the cell is often purplish blue due to abundant, rough endoplasmic reticulum needed for protein synthesis.
Serous is watery and protein-rich. Okay, this whole section really highlights the incredible specialization, from the nuts and bolts of junctions holding things together to these elaborate glands making specific products using different release methods. It's mind-boggling how much complexity there is at this microscopic scale.
It truly is. And what's fascinating, again, is that direct link between the molecular nuts and bolts, the specific proteins in a junction or secretory granule, and the overall physiological function. And when you layer on the clinical implications we've discussed, from blistering skin diseases to deafness to kidney failure, it just powerfully underscores how absolutely vital these microscopic epithelial structures are for our health. Having this deeper understanding really gives you a new lens to view almost any disease process.
Definitely. So one last area. These epithelial tissues, especially the ones forming linings, take a lot of wear and tear. Are they static or do they get replaced? Let's talk about renewal.
Yes, epithelial renewal. They are incredibly dynamic. Most epithelial cells, especially those on surfaces exposed to the environment or harsh conditions, have a surprisingly finite lifespan. They are constantly being replaced throughout our lives.
Constantly replaced? How fast?
It varies, but some are remarkably fast. We talk about continuously renewing cell populations. Think about the lining of your small intestine. Those cells are completely replaced roughly every 4 to 6 days.
Every week. Basically a new gut lining.
Pretty much. Your skin epidermis is also constantly renewing, though a bit slower, maybe around 47 days for the whole process from base to surface sloughing off.
So how does this replacement happen? Where do the new cells come from?
The replacement is driven by populations of dedicated adult stem cells. These stem cells reside in protected locations within the tissue, often called niches. For example, in the base of the intestinal glands, crypts, or in the deepest layer, stratum basal, of the epidermis.
Stem cells hiding out.
Exactly. These stem cells divide. Some daughter cells remain as stem cells to maintain the pool, while others commit to differentiating. These committed cells then multiply, differentiate into the specific functional epithelial cell types needed, and migrate upwards or outwards to replace the older cells that are eventually shed from the surface.
A continuous production line, from stem cell to functional cell to being sloughed off.
Precisely. It's a beautifully regulated process, ensuring the integrity and function of the barrier is maintained despite constant wear and tear. There are even specific molecular signals, transcription factors, that determine cell fate, like whether an intestinal stem cell daughter becomes an absorptive cell or a mucus-secreting goblet cell.
Amazing. So our skin, our gut lining, they're constantly rebuilding themselves.
They are, and this inherent capacity for renewal is also why these tissues can often repair themselves relatively well after injury. Some other epithelial tissues, like in complex glands or the liver, are more stable populations with slower turnover, but even they often retain a significant capacity to proliferate and regenerate if damaged.
This constant turnover, it really highlights the dynamic nature of our biology, doesn't it? That
Yeah.
self-healing power.
Absolutely. This final piece really brings us full circle. That continuous renewal, powered by stem cells, showcases the body's remarkable ability for maintenance and repair. It connects back to everything we discussed. The cells need to be properly formed, with the right polarity, junctions, and attachments to function during their lifespan before being replaced.
And you mentioned different membranes earlier, mucus and serous. How do they fit in?
Right, those are functional classifications of epithelial linings. A mucous membrane, or mucosa, lines cavities that connect to the outside world. Think your respiratory tract, digestive tract, urinary tract. It typically consists of the surface epithelium, which often contains mucus glands like goblet cells, a basement membrane, an underlying connective tissue layer called the lamina propria, and sometimes a thin layer of smooth muscle. Its surface is kept moist by mucus.
Mucosa lines tubes open to the outside, like the gut.
Exactly. In contrast, a serous membrane, or serosa, lines the closed body cavities, the peritoneal cavity around your abdominal organs, the pleural cavity around your lungs, the pericardial cavity around your heart. It consists of a simple squamous epithelium called mesothelium, its basement membrane, and a thin layer of connective tissue. Serous membranes don't usually contain glands, and the fluid on their surface is thin and watery, serous fluid, acting as a lubricant to allow organs to slide past each other.
The serosa lines closed cavities inside, like around the heart.
Yeah.
Different structure, different fluid, different job.
Precisely. It just emphasizes again how the body adapts this fundamental epithelial tissue type, modifying its structure, secretions, and associated layers to serve diverse functions in different environments.
Wow. Okay, what a truly deep dive that was into epithelial tissue. We've really covered the gamut, haven't we? From the basic structure, how we classify them,
To those intricate molecular details of the apical, lateral, and basal domains. We explored microvilli, ciliomodal, primary, and nodal, and those absolutely critical cell junctions. Tight junctions, desmosomes, adherence junctions, gap junctions.
right? And the basement membrane, its components, its functions, how cells attach via hemidesmosomes and focal adhesions, plus the whole world of glands, different secretion types, and finally, that constant renewal process.
You should now have a really solid foundational understanding of these vital building blocks, pulling directly from the detailed insights in Histology, a text and atlas. We've seen the tight links between structure and function, and importantly, the clinical relevance when these structures fail.
It really puts things into perspective. So here's a thought to leave you with. Next time you feel a paper cut starting to heal, or maybe even just take a breath and feel your lungs expand, take a second to consider those millions upon millions of epithelial cells.
Working constantly, in perfect coordination, renewing themselves, forming those essential barriers and linings, sensing their environment.
What other hidden microscopic wonders do you think are working tirelessly, unseen, inside your body right this very moment?
It's definitely something to ponder. Keep that curiosity alive. Keep exploring.
Absolutely. Thank you so much for joining us and being part of the Deep Dive family. We really hope this gave you some valuable insights, maybe a few aha moments. We'll catch you on the next Deep Dive.